Hello, so I want to go over this paper called Iron Regulatory Proteins, or IRP, Iron Responsive Elements, their signaling pathway in human neurodegenerative diseases. So, for those who are not fully aware, you have a protein in your body called the Iron Regulatory Protein. And let's get the Wikipedia. So this is your iron regulatory protein. And so what this protein does is it ends up binding with what are referred to as iron responsive elements. So these iron response elements are on the RNA, the messenger RNA that encodes for your proteins. So you have DNA that gets transcribed into RNA, and that RNA gets translated into the final protein product. So these iron response elements are on the mRNA. Now, granted, they're coded for on the DNA as well, but you are only getting these structures on the mRNA. And so what ends up happening is this protein ends up binding with the iron response elements. And depending on where it binds, it will determine whether or not that protein will be upregulated or downregulated. And so here we can see a small video that is describing the process, if it will, load. Sometimes this stuff just does not decide to load on here. Okay, I'll just start it. So this is an example. So here we have the mRNA that is in this secondary structure. This is the iron response element, and this is the iron response element protein. Okay, so, the iron regulatory protein. So these iron regulatory proteins and the response elements, the IREs, this signaling pathway in humans play a role in neurodegenerative diseases. So, without further ado, let's begin. And this might have to be split up into multiple videos. Anyways, the homeostasis of iron is vital to human health. And iron dyshomeostasis, or falling out of homeostasis with iron, this can lead to various disorders. So iron homeostasis is maintained by the iron regulatory proteins, the IRP1s and the IRP2s. So there's not just one of them, there are two of them in humans. And there's also the iron responsive element signaling pathway. The IRPs can bind to RNA stem loops containing an IRE, and remember these stem loops are going to be this. So here we have a stem and we have a loop here. So basically what you have is base pairing that is happening along the stem, and then you have a loop. And this stem loop is a pretty common technique that mRNA will employ to create its secondary structure. So anyways, this IRP can bind to the RNA stem loop containing an iron response element in the untranslated regions. And the untranslated regions, these are, so when you have your mRNA, and let's just look it up so that we have a good visual. So when we have an mRNA, we have the sequence for the mRNA in your cells. And this came from the DNA that got transcribed into mRNA. And there are some modifications that happen to it. For one, we have this 5' cap that is added onto it. And then we also have this poly A tail that is also added onto it. Don't worry too much. This is just out of the code, out of the 4 base letters that mRNA will use, they just have a long list of A's. Now, so you have the actual coding sequence that makes the actual protein. So here you have your start codon, and then you have a stop codon, and there's a million variations of this, but only this area is going to turn into or transcribe or encode for the protein that it wants to encode. So you can have these iron response elements in what are referred to as the 5' untranslated region, and this is the 5' side of the mRNA, so that's why it's called the 5' untranslated region, or you could be in the 3' untranslated region over here. And so depending on which side it's on, it'll change its effects. So, continuing, these IRPs can bind to the RNA stem loops containing an iron response element in the untranslated regions to manipulate translation of target mRNA. However, iron can bind to IRPs, leading to the dissociation of IRPs from the IRE. So what's happening is, if you have iron within the cell, the iron itself will then bind to these iron response or iron regulatory proteins 1 and 2, which then prevents it from being able to bind to these iron response elements. And so there will actually be a good video or a good figure down here that we can look at later. So, however, iron can bind to this, and then the dissociation or it falling apart of the IRPs from the IRE and alter translation of target transcripts. Okay. Recently, an IRE is found in the 5' untranslated region of amyloid precursor proteins. So, there is an iron-responsive element that is found in the 5' untranslated region of amyloid precursor protein, so APP. So, if you are not aware, amyloid precursor protein is one of the proteins, or it is the protein that when it is cleaved in a particular way, it can form amyloid beta plaques. And these amyloid beta plaques can basically, they're crystals of the protein, and then this can result in diseases such as Alzheimer's. So, this iron response element is found in the amyloid precursor protein, and it is also found in the alpha-synuclein transcripts, and we'll go over a little bit more of this later. So the levels of alpha-synuclein, APP, and amyloid beta peptides, as well as protein aggregation, can be downregulated by iron-responsive proteins, but are upregulated in the presence of iron accumulation. So if you have a lot of iron, then you start producing more of these amyloid precursor peptides or proteins. So therefore, inhibition of the IRE-modulated expression of APP and alpha-syn or chelation, chelation is when you have a chemical that will bind to the iron. So you can have chelation of iron in patients' brains. This has therapeutic significance to human neurodegenerative diseases. Currently, new pre-drug iron response element inhibitors with therapeutic effects have been identified and are at different stages of clinical trials for human neurodegenerative diseases. Although some promising drug candidates of chemical IRE inhibitors and iron chelating agents have been identified and are being validated in clinical trials for neurodegenerative diseases. Future studies are expected to further establish the clinical efficacy and safety of iron response element inhibitors and iron chelating agents in patients with neurodegenerative diseases. So here's the background. So the roles of iron in hemoglobin formation and oxygen transport have been linked to human health and disease. So iron is important in the functioning of many prosthetic groups, including the heme group, and this is the chemical group that gives hemoglobin its ability to hold on to oxygen, and also these iron-sulfur clusters. So iron, you might have heard of fool's gold. So fool's gold is iron and sulfur that's chemically bound to each other. So regardless, there are small bits of fool's gold that are able to participate in reduction oxidation reactions, or they're able to add one or remove one electron at a time, which is extremely valuable for enzymes because then it allows them to do these transferring of electron reactions in the body. So it helps catalyze reactions. And also, an iron depletion can contribute to anemia. So if you do not have enough iron, you can have it contributes to anemia. However, excess iron can promote the generation of deleterious reactive oxygen species. So you might have heard of free radicals, right? And why we need to have antioxidants in our system. Well, these reactive oxygen species, a lot of them are coming from the labile iron that's in your body. So if you have too much free iron, it can react with oxygen and it can react with the hydrogen peroxide in your body to create free radicals. Anyways, this is linked to both hemochromatosis and thalassemia. So hemochromatosis, I'm not familiar with what that is, so iron overload, so yeah, it's basically iron overload and thalassemia, which I don't know what, it's basically when the hemoglobin is not able to carry as much oxygen, and so these are an example of thalassemia blood cells versus normal blood cells, where they have lost a lot of the color. Okay, so the molecular mechanisms of iron metabolism in humans have been extensively studied. Ferrous iron, and ferrous is iron 2+, so it's an iron atom that has lost 2 electrons. that is absorbed from the intestinal lumen into the enterocytes. These are just the cell surface. What would you? I'll just look it up. So the enterocytes are these cells that are in the intestines that have the brush border cells on top of them. So this is an enterocyte. So these are participating in absorbing the minerals and absorbing the nutrients out of the food you eat. Anyways, ferrous iron that is absorbed from the intestinal lumen into the enterocytes can be exported from those enterocytes into the bloodstream through ferroportin. This is one of the cell surface transporters. And this is inhibited by hepcidin or facilitated by hepastin and ceruloplasmin with peroxidase activities. Okay. So the iron in bloodstream can be captured by transferrin. So this transferrin, this transferrin is a protein that can bind to iron 3+, and helps protect your body from that iron while it's being transported from one cell to another. So anyways, the iron in the bloodstream can be captured by transferrin in the ferric state and transported to peripheral tissues. And in peripheral tissues, the iron-loaded transferrin will be recognized by the transferrin receptor, or the TFR, on cell membrane. So followed by the receptor-mediated endocytosis. So basically, the receptor finds the transferrin and then will suck up that transferrin into the cell. Let's see. And so once it's taken in the acidic endosome, the iron will be dissociated from the transferrin and released into the cytoplasm through divalent metal transporter, or DMT1, after reduction up to its ferrous state by an STEAP family reductase. So this reductase in the STEAP family will add an electron to the ferric ion and then turn it into the ferrous ion. And since a ferrous ion is divalent, it's a 2+, it's in the 2+ state, it can then be transported by the divalent metal transporter 1. So and I should also mention that this, I believe DMT1 and transferrin and perhaps some of these other proteins are also regulated by this iron regulatory protein. Let's see. So in the cytoplasm, free ferrous iron can be immediately used as a cofactor for enzymes such as tyrosine hydroxylase, or taken up by mitochondria through mitoferrin for synthesis of iron-sulfur clusters and heme groups. So the iron can be taken up into the mitochondria, where it can form into these iron-sulfur clusters, or the hemoglobin, or the heme group, which is a part of the hemoglobin molecule. And this is indispensable for mitochondrial function. Considerable amount of iron in cells can be sequestered and stored in cytosol ferritin. So ferritin, in contrast to transferrin, ferritin is going to hold on to iron and store it, but it's not transferring it between the cells. So it can happen, this cytosolic ferritin can take up a lot of the iron, or also the mitochondrial ferritin can take it up in the ferric state. So this is also in the iron 3+ state. So excess iron can be exported into the bloodstream through these ferroportin, or FPN, I forgot what that is, one second. Ferroportin, yes, so it was ferroportin. So it can be transferred into the bloodstream through ferroportin to form ferric iron transferrin complex again for iron redistribution. So in this particular case, that transferrin could actually be used to, excrete the iron as well. So you might not necessarily, you might have iron overload. So you want to get it out of the cells and possibly, excrete it in some way. Anyways, a substantial amount of iron can be absorbed into the brain, mainly in the substantia nigra pars compacta, also known as the SN. And the iron in the bloodstream can be transported across the blood-brain barrier through brain capillary endothelial cells, or BCECs, through the transferrin receptor and the DMT1 ferroportin pathway. So remember, this is the transferrin and transferrin receptors, which uses endocytosis, and this is the DMT ferroportin pathways, which this one is just pumping it through the cellular membrane. So the circulating iron transferrin complex can be captured by the transferrin receptor on the brain capillary endothelial cell membrane. It can be internalized through endocytosis, or it could be released to the BCEC cytoplasm through the DMT1 and exported into the brain interstitial fluid through the ferroportin. So in brain, iron plays multiple physiological roles, including neurotransmitter synthesis, neuron myelination, and remember the myelin sheath. This is an insulator that is on the axons of your cells, and this iron actually plays a very important role in the proper, well, I shouldn't say iron does, but the transferrin in the brain plays an extremely important role in the differentiation of the oligodendroglial progenitor cells, turning them into the fully mature oligodendroglial cells. And those oligodendroglial cells are very important at remyelinating or adding new myelin sheath onto damaged neurons. And so that requires transferrin, which proper expression of transferrin requires sufficient iron in the body. And if you don't have sufficient iron in the body while you are developing, you can actually have a huge reduction in cognitive function as a result of that. So let's continue. So there's also mitochondrial function and energy generation in the brain as well. So iron homeostasis in the brain is precisely controlled and dysregulated brain iron homeostasis from either iron overload or deficiency. This can lead to brain disorders. So brain iron deficiency, or BID, can disturb brain development and function. BID can be associated with pathogenesis of brain disorders, including attention deficit hyperactivity disorder and restless leg syndrome. So BID can be associated with pathogenesis of brain disorders, including attention deficit hyperactivity disorder and restless leg syndrome. So, I mean, I remember when I was younger, I actually was a little bit anemic, and then I did believe that I had some RLS. Well, for one, my legs could not, like, they were restless, for lack of a better word. So I can definitely see this being true. So, and also the hyperactivity disorder, you'd think that if you didn't have enough iron that you would, be lethargic, but apparently the inability to concentrate and hyper-focus on way too many things at the same time, this can be associated with iron deficiency. Anyways, the iron overload in the brain can also be a pathological factor for brain disorders, including Alzheimer's disease, Parkinson's disease, and other human brain disorders. So this dysregulation of iron, and actually the accumulation of iron in the brain, is associated with these neurodegenerative disorders, Alzheimer's disease, Parkinson's disease, and other human brain disorders. So, and that could be even if you are anemic, like you might not even have enough iron in your body, but if it gets localized into the brain, that can cause us even worse effects, right? Anyways, modulation of iron homeostasis in the IRP-IRE signaling pathway. So this is the iron regulatory protein and the iron responsive element signaling pathways. So iron homeostasis is elaborately regulated, although some control mechanisms exist at the transcriptional level. So this is the transcriptional level is going from DNA being transcribed into RNA, right? So there is some regulation that is happening between DNA and RNA, but more of it is being done through the mRNA to the protein. Anyway, so it can happen at the transcriptional level. Also, at the absorption and the transportation and the storage of iron are meticulously modulated at the translational level by the iron regulatory protein, the IRP, and the iron responsive element, IRE signaling pathway. So even though some of it is being done in the DNA, a lot of it is being done at the RNA level. And this, to me, makes sense because it's something that you might need it to be shifted very quickly, right? So this is something that your body has decided, I need to act quickly when something like this happens. So I could see something important here is, let's say someone basically gets gashed, and so they lose a lot of blood immediately. Well, they're going to have a huge iron deficiency at that very moment, right? And so you might want to get the body immediately in gear to store and save iron if you have a giant gash in your body. So that could be, that could be for one of the most important nutrients. It makes sense that you'd use one of these faster regulatory mechanisms. Anyways, IRP1, which is 90 kilodaltons, and IRP2, which is 105 kilodaltons, are two known RNA-binding proteins, and their inducible interactions with the IRE function their inducible interactions with the iron response element. This function to control the translation of ferritin and ferroportin mRNA and the stability of transferrin receptor mRNA. So ferritin and this ferroportin, its translation is controlled by IRE, and its stability affects the transferrin mRNA. So, sorry, the transferrin receptor mRNA. Briefly, the IRPs can control iron metabolism through binding to specific non-coding sequences known as the iron response elements within the untranslated regions of mRNA transcripts. The IRE are 30 nucleotide long RNA motifs containing the CAGUGN sequence, which is the classic IRE motif, and this can form special stem loop structures, which is what I showed you earlier. IREs can be present in either the 3' untranslated region or 5' untranslated region of the target mRNA. Transcripts with IRE motifs in their 5' untranslated region include the ferritin H and L subunits, ferroportin, and aminolevulinic acid synthase. Don't worry too much about this. So, whereas the target mRNA with the IRE motifs in the 3' untranslated region include the transferrin receptor and DMT1. And the details of IRP-IRE signaling pathway translation modulation mechanisms are illustrated in Figure 1. So here we have, I don't know if I should have clicked that, but 1 second. So here we have the iron regulatory protein. So these green things are the iron regulatory proteins. And so normally, let's say this is your mRNA. Your mRNA will have the iron responsive element somewhere on it. And in this case, it's in the 5' untranslated region. Okay? And so normally, you have synthesis of this protein. And what happens is, when the iron responsive element binds to this when the iron regulatory protein binds to the iron responsive element in the 5' region, it will stop translation. It prevents translation from happening, and so you do not get your final protein. However, when you have iron, the iron will bind to the iron regulatory protein, which then knocks it off of the iron responsive element, and then you have translation and you finally get the protein. So in this particular set of circumstances, iron will promote the production of protein. Okay, so the more iron, the more protein. Now, there are different mRNAs where it is in the 3' untranslated region. So what happens is, if you have a lot of these secondary structures in the 3' untranslated region, that can signal to the body to destroy, signal to the cell to destroy this mRNA, and then you don't get any protein. And so you get endonuclease cleavage, or you get destruction of this nucleotide. Now, so I should say polynucleotide. And if you have your iron regulatory proteins, though, they will bind to these IREs and prevent this endonuclease activity. And so the mRNA stays, and the mRNA persists in the cell, which then gives it time to encode for the protein. Now, if you have iron in your body, that will actually kick off these iron regulatory proteins from the iron responsive elements, and as a result, you end up having this being cleaved. And so what you can then say is iron for some other proteins or for some other mRNAs, if you have more iron in it, you get more destruction of that mRNA, and more destruction means you have less protein. So in this case, iron means less protein, and in this first case, iron means more protein. So you could imagine iron, you want both up regulation of some things and down regulations of other things, and so your body knows what to do based on the location of the iron responsive element is. So that would be a different logic gate, right? So presence of iron, if true, then do this. So that would be the logical progression of this particular mRNA. Now going back, you know what? I think inspect. Delete. Sorry, I hate. So I think I will leave it at this for now, but I'm going to do other videos on this. So yeah, subscribe if you want to learn more about this and how these iron regulatory proteins play a role in neurodegenerative disorders. And hopefully I will see you later.